baby. You know that I miss you. I wanna get with you tonight, but I cannot, baby girl, and that's the issue. Girl, you know I miss you. I just wanna kiss you, but I can't right now, so baby, kiss me through the phone.
The purpose of the experiment is to determine if change in pH affects the rate of photosynthesis.
We hypothesized that the change in pH does affect the rate of photosynthesis because water has a neutral pH of 7, and photosynthetic reactions need water to occur. At acidic or basic pH, proteins that catalyze the photosynthetic reactions, such as photosystem 1 and photosystem 2, are denatured, meaning their tertiary structures unfold. Without functioning proteins, photosynthesis cannot occur. So, at pH of 4 and 10, photosynthesis will slow or stop, shown through a small change in absorbance. The rate of photosynthesis will be bell curve shaped, the optimal pH of 7 in the center having the highest rate and the rate decreasing steadily at lower or higher pH.
We used 3 experimental tubes, each containing DCPIP, H2O, chloroplast, and a buffer solution. 3 trials were done for each of the 3 buffer solutions. In each trial, the experiment was kept exactly the same for each tube. We measured the initial absorbance and then put the tubes 30 centimeters from the light at its maximum intensity. We kept each tube in each trial under the light for 6 minutes, and the optimal time was determined through a first trial without buffer. We then measured the absorbance after 6 minutes for each tube. The absorbance of 3 controlled test tubes were also measured, 1 for each buffer, without chloroplasts in solution. This is due to the fact that the buffer solutions themselves are colored, and because color affects absorbance, we measured the absorbance of the colored buffer solutions alone and then subtracted these absorbance values from our absorbance values for the experimental test tube. Thus, we accounted for the fact that the buffers themselves absorb light, and if we had not, this would have been a confounding variable. The initial absorbances were subtracted from the final, and the control absorbance value subtracted from each to obtain the difference in absorbance for each buffer solution.
Figure 1 shows that the buffers of pH 4 and 10 have very little absorbance, meaning that very little photosynthesis occurred. The solution with pH 7 buffer had the highest absorbance, nearly 5 times greater than the buffer solutions of pH 4 and 10. Chloroplasts in pH 4 buffer produced least amount of product and absorbance of 0.06. Chloroplasts in pH 7 buffer created most product, measured at an absorbance of 0.35. Chloroplasts in pH 10 buffer created very little product, with an absorbance of 0.07.
Change in absorbance represents DCPIP reduction. The larger the difference in absorbance after the chloroplast exposure to sunlight, the more DCPIP was reduced, and the more photosynthesis occurred overall. In a buffer solution of pH 7, high rates of photosynthesis occurred, which supports our hypothesis that pH 7 is optimal for photosynthetic enzymes as there are equal concentrations of hydroxide and hydronium at a neutral pH. Our results also support the claim that photosynthesis rates are low in solutions of very acidic or basic pHs. According to Pollander, a hydrogen bonding network plays a catalytic role in photosynthetic oxygen evolution, which occurs when H2O is oxidized into oxygen. This reaction occurs along with the reduction of NADPH to NADP+, and so hydrogen bonding is vital to both the tertiary structure and the function of photosystems 1, 2, and ATP synthase. As the pH becomes too high or too low, these enzymes are denatured because excessive charged hydronium or hydroxide ions disturb the hydrogen bonding systems and the enzymes denature. More specifically, the active site may be altered so that substrates can no longer bind. As a result, rates of photosynthesis decrease. Our results support this claim.
That's it?
I broke up with my ex-girl. Here's a number.
What's that shit say?
Psych! That's the wrong number!